Bismillahirrahmanirrahim. Today I would like to share the knowledge about what is the PCR or PCR procedure or the conventional PCR amplification. So what is the PCR? And this one is actually the polymerase chain reaction. Indispensable technique, and it is the technique which is actually to amplify a single or few copies of DNA to thousands or millions of copies. So in 1983, Carey Mullis, that is name of the scientist who developed the PCR technique, and in 1993 he was awarded a Nobel Prize. So this one is the important techniques. That is why he was awarded a Nobel Prize. And this is actually the sketch of this that the PCR. And you can see here that this is the template DNA, and this one is the wanted gene, and which is inserted into this template DNA. And this template DNA actually in the first cycle you can see it experience that 2 copies. Then in the another cycle it is divided into 8 copies, then 16 copies, then 32 copies. After the 35th cycle its copies are 2^36. It means that 68 million copies of the single DNA which is required and which is wanted DNA. So this is one is the this overview or the sketch of that this PCR technique. So you can see here these are the main steps of that PCR. That is the initial denaturation, where 95 degrees centigrade for 3 minutes. In the case of the hot starch, it may be prolonged up to 10 minutes. This one is for 1 cycle. Another is that the denaturation, that is 94 degrees centigrade for 10 seconds. Then annealing, that is 50 to 60 degrees for 20 seconds, 30, and then extension, that is the 72 degrees centigrade for 30 seconds. So these 3 steps which is for the which are for the 35 cycles, and then final extension 32 degrees centigrade for 5 minutes for 1 cycle, and then the final holds that that is at the 25 degree hold for the 1 cycle. So these are the steps of that that the PCR. And you can see here that is the polymerase chain reaction. This is the single that DNA which is then in the denaturation, then annealing. You can see here that the DNA break into different parts, and then the extension, that is the recoiled DNA is multiplied in millions copies. So what is the principle of that technique, which is you can see here in an in vitro method for the exponential amplification of the target portion of the template DNA, which involves incorporation of the nucleotides by DNA polymerase during the thermal cycling. So what is the procedure for doing the PCR? This is the short or is very brief procedure to follow the PCR techniques. So first of all, you label the PCR tubes for the samples and controls, and it is very important you should label all the PCR tubes. Then thaw the PCR reagents and prepare the PCR reaction mixture, and add that in the lab. Then aliquate the reaction mixture in the individual PCR reaction tubes or wells. Open the PCR machine software and edit the run parameters, and then place the sample tubes in the thermal cycler, close the lid and run the program. And on the PCR machines then after putting the PCR sample tubes, after that completion of the PCR, then you remove the tubes and proceed the sample for agarose gel electrophoresis, and or otherwise you should store the PCR product at the -20°C. So these are the references. You can open these link and you can follow the detail of that procedure. So this video is actually that the helpful for the initial or that the researcher who are trying to learn about the PCR reaction techniques. So this is the short and brief video, and if you have any query or any questions or any suggestion, you can put at my YouTube channel. I will I try my best to give you the response as soon as possible. Thank you so much.